Potassium is important in the analysis of pigments because it is indicative of the way some specific class of pigments, the lakes, they were made. Lakes basically are made by plants, so basically crushing leaves from specific plants, flowers, leaves. And in order to extract the colorant from these leaves, from these plants, generally it was used an alkaline solution, a base. From antiquity, the bases that they have, there were few of them, could be based on sodium, could be based on potassium. And so from potassium you have alum, potassium alum sulfate, or you can use potash, potassium hydroxide. While potassium will not be the main component of the colorant in this lake, though the presence of potassium can distinguish different manufacturing process. Also, for example, we will see potassium also is added in iron gold inks in order to make them more fluid and to preserve the inks better. Potassium, it's not the main protagonist for the pigments, but definitely play a role. In this class, we'll see how to identify potassium. As usual, we will look at first at the characteristics of its X-rays, and we will discuss what is the best experimental setup for our spectrometer to detect the tiny amount of potassium. This is also important because we are looking at traces of potassium in these lakes. Generally, potassium is not the main component, so there will be just traces. So we need to use our spectrometer at its best in order to detect this tiny amount of potassium. Potassium number 19. Potassium is the X-ray table, so it has a K-alpha peak at 3.3 and the K-beta is at 3.6. So you see now, basically with potassium, the K-alpha and the K-beta are far apart. We have 0.3 keV of distance between the two peaks. Now we expect them to appear in our spectra as separate peaks. Remember, when we did analyze, for example, here chlorine, you see the difference here is just 0.2 keV. In particular, in sulfur, we see that the difference between the K-alpha and the K-beta is just 0.1 keV. This distance is so small that is less than the resolution of most of these spectrometers for X-ray, these handheld spectrometer. The method I suggest to detect potassium in this small amount is the no-filter setup. Indeed, if we look, this one is the HDPE spectrum, the spectrum that we get when we just have a tablet, a plate of high density polyethylene under the spectrometer. In this case, you see this area is the area that we already discussed for the other light element like aluminum, sulfur. This area here, you see there is these big peaks. These are from the rhodium target of my specific tube. We cannot use this area, but then we see this area here over basically 3 keV. This area is pretty flat, doesn't have peaks coming from these elements inside the X-ray tube. When it comes to 3.3, we are over here. Basically, we can use this no-tube filter setup to detect peaks that are in this area. definitely it makes sense to use the no-filter setup to detect potassium. We go in the lab and we start our first experiment. The easiest source of potassium is alum. This one will be potassium aluminum sulfate. So we test it. So this one is the alum. So alum is this short name to indicate this potassium aluminum sulfate, material used from antiquity in order to get these alkaline solutions that are important to extract the colorant, in particular for the manufacturing of lakes. Well, there it is. So this is the spectrum that we get with no filter setup from alum. We see the 2 peak K alpha and K beta of alum. The potassium is over here, so you see the 2 peaks are far apart now, we can easily distinguish them. And then also we see sulfur over here and aluminum over here. And the count you see here is 50,000, so we get a lot of signal from potassium. This is indicative that we are going to see a lot of the potassium even when it is at low concentration. Okay, let's move in the lab and try something else. We test now still the grain. This one is a lake. We have some plants, so could be flowers, could be the leaves, and from these plants we extract the colorant. The colorant generally is a complex molecule that has to be then fixed to a substrate. In all this process, so the extraction or the fixation into a substrate, potassium can play a role and be present, and so we can detect it. We must say so that it's not potassium that is the origin of the color. Potassium is not the origin of the color, but it's could be present, indicating the manufacturing process. Let's test it. There it is, still the grain, the pigments we just analyzed, and this is the spectrum. Still the grain has potassium, so we see the peak over here at 3.3, but of course is much less than what we saw in alum. What we see here, there is much more of calcium, of course, because also from the paper, from the support by the material, this lake, the substrate of this lake, potassium is present in there, because still the grain is made by crushing the berries of this plant, the buckthorn berries. They are crushed, and these crushed berries, they are put in an alkaline solution containing potassium. This alkaline solution with the potassium can crush better the structure of the cells of these berries, so that the colorant can better go into the solution and then can be used to be fixed. Doing XRF spectroscopy of the grain, we see potassium, so we have distinguished, we have traces, information about its manufacturing, because other products can be done with other elements. So instead of using potassium, you can use sodium to make an alkaline solution. This is the information we can take. Let's try another lake. The leaves of the Indigofera tinctoria are crushed and steeped into a solution containing potassium hydroxide in order to precipitate the colorant from this indigo plant. Let's see if we can see the potassium in it. So we just test indigo, and from indigo we see the presence is the blue one, spectrum blue one, we see the peak of potassium again. The black one is the background, basically, signal, the HDPA, the high density polyethylene. So we see a clear peaks of potassium. Again, this is indicative that there was an alkaline solution present for the manufacturing of indigo that was containing potassium. So we have traces of this potassium over here in this peak. Let's go in the lab and now we try another material. Ottoman 19th century manuscript. Let's analyze this iron gold ink that also has created a lot of damage in the manuscript. So we did analyze this iron gold ink and now we look at the spectrum. I'm comparing the spectrum of the paper, so is the black one. We see here the paper, the black, so for example here we have the peak of calcium, which is normal in paper. The peak basically here overlap, so the spectra of the paper overlap that of the ink, the black ink. The only difference basically is this big peak over here, which is potassium. This potassium comes from the iron gold ink that is over here. And generally we explain the presence of potassium, which is not the main component of the iron gold ink, but it could come at the time they use this potassium carbonate in order to stabilize the ink to make it stay longer. The important concept is that we can detect it, we can quantify it, so the intensity of the peak, so we can distinguish inks by their potassium content.